Welcome to today's Explainer. Get ready because we are throwing on our lab coats and stepping right into the shoes of plant scientists. We're not just passively observing nature today, folks. We are about to embark on a highly controlled botanical sting operation. Our prime target? The broad bean. We all know these little guys eventually grow into plants, but today we're going to uncover exactly how that whole process gets started.
Okay, let's dive into this. Have you ever wondered what is actually going on when a seed is just sitting there in the dirt? I mean, to the naked eye, it looks like a tiny, lifeless pebble, right? Like it's completely asleep. But here's the thing. It is actually highly alert. It is literally just waiting for very specific environmental signals to trigger its grand awakening. So, our big question for this mission is, what exact conditions does a broad bean seed need to wake up and start growing? Let's figure this out together.
To crack this case, here is our quick mission briefing. First up, the germination mission. Second, brainstorming seed needs. Third, designing the experiment. Fourth, ensuring a fair test. And finally, number 5, gathering and recording data.
Let's kick things off with Section 1, the germination mission.
Before we go any further, we need to make sure we're on the exact same page with our baseline objective. The scientific concept we are dealing with here is germination. Very simply, germination is that specific moment when a seed starts to grow into a new plant. It's this incredible, almost magical transition from a dormant, hard little shell into a living, breathing, biological organism that's actively pushing its way into the world. That exact transition is what we are trying to capture today.
Now, what's really interesting about this is the actual anatomy of our target. A seed is definitely not just a solid lump of matter. It's honestly a brilliant piece of biological engineering. Inside, it's got an embryo, which is the actual baby plant just waiting to grow. Surrounding that embryo is a food store, which gives it all the essential energy it needs for that massive initial burst of growth. And wrapping that whole shebang up is the seed coat, a super tough armor protecting the package until the timing is absolutely perfect. So it basically comes pre-packaged with its own food store and armor.
Moving right along to Section 2, brainstorming seed needs.
All right, it's time to think like detectives. We've got a whole lineup of environmental suspects that might be responsible for waking the seed up. Take a look at these potential factors. Is it water? Does it need light? What about the temperature? Does it need air and oxygen? Or is soil strictly necessary for this to happen? We can't just guess here. We have to systematically isolate these suspects to see which ones are actually pulling the strings.
This brings us to Section 3, designing the experiment.
To execute our strategy, we are setting up a 4-jar operation, and this brilliantly illustrates how we can actually test these factors. Each clean glass jar represents a totally unique environment. Jar 1 gets water and air, but no light. It goes right in a dark cupboard. Jar 2 gets water, but we restrict the oxygen, and again, no light. Jar 3, that one gets everything, water, air, and plenty of light out in a normal classroom. Finally, Jar 4 gets normal air and no light, but only a bare minimum of water to simulate really dry conditions. By uniquely isolating light, water, and oxygen across these 4 jars, we can see exactly what the broad bean responds to.
Now, the methodology for this operation over the next 2 to 3 weeks is strict. Step 1, place some moist kitchen roll or cotton wool along with 1 or 2 broad beans into your 4 clearly labeled jars. Make sure to place the seeds right against the glass so we can keep a close eye on them. Step 2, apply your assigned light, water, and oxygen conditions to each specific jar. And Step 3, observe and photograph the jars twice a week, let's say every Monday and Thursday. Now here's a crucial directive. Do not move the jars between conditions unless instructed. Consistency is absolutely key for this to work.
Next up is Section 4, ensuring a fair test.
In any scientific investigation, the rules of engagement dictate that we have to fully understand our variables. The independent variable is the thing we deliberately change, like the amount of light or water. The dependent variable is the outcome we are actively measuring, like root length or the germination rate. So the crucial point here really comes down to the controlled variables. Keeping these controlled variables, like using the exact same bean type, the same jar size, the same tissue paper, and keeping the background temperature totally identical, is the only way to ensure our final results are actually trustworthy. I mean, think about it. If one jar is way warmer than the others, we won't have a clue if the seed grew because of the light we gave it or just because it was hot.
And finally, Section 5, gathering and recording data.
Okay, so we need a very strict definition of success for an individual seed. When do we officially call it? For this investigation, germination is recorded the exact moment the first visible part of the embryo, which is normally the young root, also called the radicle, actually emerges from the seed. You're going to be watching for the seed to swell, the coat to split, and then boom, that root pops right out. When it does, we'll measure the root and eventually the shoot length using a millimeter scale.
Let's move to the data table and see how this builds into our final scientific conclusion. We're going to take all of those individual seed observations and carefully log our intel right here. By dividing the number of seeds that successfully germinated by the total number of seeds we tested in each condition, and then multiplying that by 100, we get our definitive germination rate percentage. We'll also average out the root and shoot lengths. This way, we're not just seeing if they germinated, but figuring out under which exact conditions they grew the strongest and healthiest.
Before you start this investigation, I want to leave you with a thought to really mull over. Imagine you gather all your intel, and you find out that jar 3, which is sitting out in the bright classroom light, and jar 1, which is locked away in a totally dark cupboard, both germinate perfectly. So what would that tell us about a seed's actual need for sunlight during those first few days when it is naturally buried deep underground? It's a pretty fascinating question, right? Well, it is time to set up your jars, uncover the hidden life of seeds, and find out for yourself.